I didn't even associate it with LSU until just now. Like, great.
Are you attended?
This is just the
Zero challenge. She really does. I don't think she's doing her yet. She's Karen's still. It's cheap.
Nobody's sitting there. You just have to put up with that computer being there. I'm not able to get back into it now, so I'm just gonna use that one. Yeah. No problem.
If we move some, maybe into the back. Yes.
Like my kid goes there, maybe.
Yeah. All it's gonna do is make me nervous.
I mean, unless these chairs here, so I'm gonna sit here. Just very educated, he is not.
Yeah.
Nope, not that one. Connect audio, start video. Hey Scott, can you see me and hear me? It's promising. How about now?
Can you use Skype?
He's not allowed to use Skype on his
So,
government computer.
Yeah, so, okay.
He's been trying to set this up since like 11 o'clock.
So just send him PowerPoint slide.
Okay.
She see you.
Yeah,
If she can see you, we can use maybe telephone for the regular speaker.
all right.
So just ask him to use to go to YouTube.
Yeah, that's what he said. He said he's going. We can just start. Like, I mean,
Is this a Zoom link?
well, he's with the government, so he's not allowed to use any of those softwares, and so the WebEx one is not working, and he can't hear us or see us. And so,
Can he do a phone?
yeah, but then
Did you send him the slides?
no. But I have a live YouTube stream going, and I think that's just gonna have to work. Like,
And he's getting that?
I think he should be able to see that.
I mean, the other thing you could do is send him the slides.
Yeah.
If you send him the slides, and then you could put phone also next to you, he can.
Okay.
Why don't you do that as a backup?
Okay.
Send him the slides.
Okay.
Within the government, this is over.
Park Service.
So he's wrong.
Was here.
Okay. So he should have access to the slides. Let's go. Okay. Now this. Good.
You want to? Okay. Thank you for coming to today Prospectus presentation. I'm pleased to introduce Laura Scott. And Laura is a second year PhD student in the Department of Global Environmental Health Sciences. And Laura received her Bachelor in Science in Zoology from Oklahoma State University and Master in Epidemiology in Tulane University. And Laura also received the Bailey Rocky Mountain Research Fellowship, which allowed her to conduct research on antibiotic-resistant bacteria in soils and waterways in Rocky Mountain National Park. And recently, she was awarded a student travel award from American Society for Microbiology to present her work at the ASM, American Society of Microbiology General Meeting this summer. So without further ado, I will allow Laura to present her research proposal.
Thanks. All right, so welcome everybody. Sorry for the technical delays and the late start. So I'm going to talk today about my dissertation proposal, which is titled The Characterization of Antibiotic-Resistant Bacteria, Their Resistance Genes in Soil and Water Environments, and the Factors that are Related to Their Dissemination. So just a quick outline of how we're going to go through this today. I'm going to give you a little bit of background about antibiotic resistance in general and why it's an issue. Then I'm going to describe my 3 dissertation aims, and within those give you a little bit of relevant background to tell you what my hypothesis is, show you any preliminary data I might have, talk about my expected experimental design, and then what I expect to find in some of the limitations that I know exist. And then finally, I'm going to kind of wrap it all up by the totality of my expectations and kind of what the conclusions of all of this are. So antibiotic resistance, why is it a problem? How do bacteria become resistant in the first place? Well, the most commonly thought of way is through natural selection. So sometimes bacteria just happen to have different types of proteins or that allow them to either pump toxic substances out of their bodies or out of their cells, or allow them to break those substances down, which means that when they come into contact with antibiotics, they don't die, which means that they are resistant. And so what the little graphic at the bottom shows is that the resistant ones survive, they reproduce, and then we go on and we have this problem of resistance. And that's kind of just the process of natural selection and evolution that we normally think of. But there are other ways that bacteria might actually become resistant. And one of those is through what's called horizontal gene transfer, which is a way that microbes are kind of playing this totally different evolutionary game than we are at the eukaryotic level. And what they can do through the processes of transduction and conjugation is that they can share their genes with each other. And so sometimes antibiotic resistance genes can be passed from one bacteria to another without reproduction happening. So this graphic is meant to represent how antibiotic resistance has changed and kind of come over time. So what you'll see on the top of the timeline is when resistance to a particular antibiotic cropped up. And what you see on the bottom is the year that that particular antibiotic was put on the market. And so you'll see that even with the very first antibiotic, penicillin, there were resistant isolates to penicillin before it was ever even put on the market. And with even our most recent antibiotics, which there are very few now, pretty much as soon as they come on the market, we start seeing resistant isolates to them. So antibiotic resistance is definitely a problem. Also, just to kind of back up the idea that antibiotics might be contributing to why we're seeing more resistance, so a study was done by the Center for Disease Dynamics, Economics, and Policy that showed that the more often vancomycin was prescribed, the more mortality they saw in C. diff infections. And then alternatively, the graph on the right just shows that over time, mortality and prevalence have increased for antibiotic-resistant infections. The CDC in 2013 put out like a news report saying what they thought were the most important threats regarding antibiotic resistance. And it's basically a list of pathogens that they think are the most important. The urgent threats are the ones that they think are the biggest concerns. And like we just talked about, C. diff is one of those that you see often in hospital-acquired infections. And then the other two on that list are of great importance because they're resistant to last resort drugs, meaning that there's nothing you can treat them with once they're resistant to those drugs. You'll also, on the serious threat list and the concerning list, probably see some pathogens that you've heard of before. Most of us have heard of MRSA and vancomycin-resistant Staphylococcus. It's estimated now that about 30% of the regular population carries MRSA, isolates on their skin. VRE is another common one that people talk about. And so there are a lot of pathogens of concern. So what's the public health significance of antibiotic resistance? Like, obviously, we're not all falling dead from resistant infections all the time. So something else must be going on here. So in 2010, the CDC estimated that about 2 million antibiotic-resistant infections occurred in the United States. However, a study done by Burham, Olson, and Coliff suggested for that same year that mortality was somewhere closer to 153,000 when the CDC estimated that mortality was around 23,000. So the estimates are at best not very good. Internationally speaking, the WHO is very concerned about multi-drug resistant tuberculosis and extreme drug resistant tuberculosis, meaning it's just at that point untreatable. The WHO also looks at how antibiotic resistance is changing throughout the world by what types of drugs are being requested. So higher and higher level drugs are being requested more frequently, meaning that resistance is on the rise. And also there's been a rise in colistin resistance in Enterobacteriaceae, which is a very common gut bacteria. And again, colistin is a last resort drug. So it's definitely of concern that people are getting infections to these last resort drugs. So when you think of antibiotic resistant bacteria, a lot of times we think of hospitals. People go to the hospital, they have surgery, they get these infections, and then they die. But what the data actually shows that I took from CDC, you can kind of see on the left by pathogen from 2011 to 2014 in terms of like percent isolates that they're getting from patients that are resistant. It's either staying stable or it's on the decline. And then you'll see when you combine all those pathogens that in general, like the percent of isolates that are resistant from hospital-acquired infections is actually on the decline, despite what we saw earlier, that resistant infections as a whole are on the rise. So what that means is people are getting them out in their community, out in the environment. So what does that mean? Antibiotic-resistant bacteria out in the environment. Part of what we're going to talk about today is that they're kind of inefficient to measure. And one of the answers to that might be that flow cytometry could be a better way to do it compared to the traditional method. There have been very few studies actually looking at antibiotic-resistant bacteria in environmental media. However, the chart on the right is from a study that was done in Antarctic and other glacial studies. And the colors might be a little bit difficult to discern from here, but basically what it shows is that the isolates found more frequently were found in glacial areas that were closer to places with higher human densities, suggesting that the presence of humans has something to do with the frequency with which we see antibiotic-resistant bacteria, which kind of makes sense. So on the flip side of that, antibiotic resistance genes, or ARGs, as you might hear me call them for the rest of this presentation, have been much more widely studied. And that's partly because they're more efficient to measure. It's cheaper, it's more high throughput because you can use genomic methods. However, a lot of the studies don't really focus on, the studies do focus on likely sources. So places where we would expect to find these resistance genes, like wastewater treatment plants. If we think they're coming from people, then when people go to the bathroom, that's probably where we're going to find bacteria that are resistant. Or looking in hospitals, again, places where we're likely to see them. So there have not been very many studies that have been done looking at places where it's less likely to find them, or kind of along a human gradient. The other problem with looking at just resistance genes is that they don't tell us anything about bacterial viability. And ultimately, if we want to know something about risk and whether or not these resistant bacteria that are out in the community or out in the environment are of risk to people, we need to know whether or not the bacteria are alive. So there are definitely some gaps in knowledge. So are antibiotic-resistant bacteria and or genes ubiquitous throughout the environment? Are they in pristine places already? Are they everywhere? Are they everywhere in equal concentration? We have no idea as of right now. How are these resistant bacteria and these resistance genes getting into the environment? How are they being disseminated? How do they move around? What factors contribute to that? How much does human activity account for that? Is there an easier way for us to measure resistant bacteria from environmental samples so that we can maybe speak to risk further down the line? And then what factors determine, again, kind of how these things move within the environment? So you're going to hear me talk about national parks because that's going to be a little bit of the focus of my research. So I just wanted to give you a little bit of background about why I study these things in national parks. Well, national parks have really fantastic examples of human gradients. So the picture you can see up at the top left is in Rocky Mountain National Park at a place called along a road called Trail Ridge Road, which is pretty popular. And what you'll see is that one person way up at the front probably saw a stick that they thought was an elk, so they stopped in the middle of the road, and now traffic's all backed up, and you've got thousands of people in this tiny amount of space. But the next picture over to the right is Rogue Glacier, and it's estimated that maybe 10 people visit it per year because it's so difficult to get to. So there's a stark difference in traffic within, a 1 square mile area and, less than 10 people seeing a wide range of space per year. Also, national parks are protected environments. What that means for a study like this is it might make us less likely to run into different types of contamination confounders. And parks also have access to a lot of data. A lot of people are doing a lot of very different research in parks, and all that data ends up getting shared. So some of the examples that I provided here are, there's someone doing a 30-year butterfly study. How might insects or birds be carrying these resistance genes or resistant bacteria through the park as they do their yearly migratory patterns? Or the larger mammals, like they get ticks. How are those moving around? How does that contribute? How do physical and chemical parameters in the water contribute to resistance genes and resistant bacteria? And then finally, people are doing ice core studies in the national parks, particularly in Rocky, and that can ultimately maybe inform us a little bit about how antibiotic resistance has changed over time, which would be really interesting. So kind of in conclusion, antibiotic-resistant bacteria are a serious public health threat identified both by the CDC and the WHO. Antibiotic-resistant bacteria and their genes in the environment have not been very well described. Measuring the bacteria themselves can be pretty costly and inefficient, difficult to do. Most studies focus on sites of where they expect to find them, so places with human waste. The factors related to the dissemination of resistant bacteria and resistance genes have been pretty understudied. There have been some studies looking at the correlations between resistance genes and heavy metals. I'll talk a little bit more about that, but there's not a lot of strong correlative data there yet. And very little data exists on how these resistant bacteria and these resistance genes might be changing in the environment over time, because we're just now starting to look at it. And so just as I showed you with that timeline earlier, the resistance to drugs have changed over time and how quickly the drugs and the resistance change. So it's just as likely that these things are changing in the environment over time as well. So I'm going to quickly kind of highlight my general hypothesis and my 3 aims, and then I'll go into each one of them with a little bit more detail. So my general hypothesis is that human activity and human density are spatially related to the presence of antibiotic-resistant bacteria and their genes in national parks. So my first aim is to develop a high-throughput method for quantifying resistant and susceptible bacteria to both bactericidal and bacteriostatic antibiotics from environmental samples using flow cytometry. My second aim is going to be to sample and analyze soil and water samples across a human impact gradient in 2 different national parks, looking for antibiotic-resistant bacteria and specified genes. And then my third aim is going to be investigating the genotypic and phenotypic effects of 5 heavy metals on the antibiotic susceptibility of both gram-positive and gram-negative bacteria. So the first aim, which again is to develop this high-throughput method for looking at antibiotic-resistant bacteria using flow cytometry. So what is flow cytometry? You might not be super familiar with it. The way it works is you put your sample in and it's exposed to a laser light source, and then it's basically the shadow that it casts is measured, and that's one way of looking at how your sample turns out. The other way is to tag your sample with a fluorescent probe. And so what the graph kind of at the bottom middle shows is that with fluorescence, you've got this excitation wavelength and then this very highly expected emission wavelength. So we can see with some specificity when we tag something, if we see that emission wavelength, we know that what we're looking at is what we expect we're looking at. And the way that kind of works in the context of flow cytometry is when all of us go shopping at Claire's, is that you walk through the store and that's how they count people. They've got these bars, which you can see kind of over on the right, and they are lasers. And so when you break the boundary of that laser, every time that boundary is broken, it counts you. And so that's basically the same way flow cytometry works. And so that's how we would be able to count the bacteria that get passed through the flow cytometer. So why flow cytometry and not something else? Well, flow cytometry comparatively is very fast, and that's what the graph or the figure on the right shows. So to summarize that quickly, for 96 samples could potentially get all of your samples completely read and analyzed in about 6 to 12 and a half hours using flow cytometry, as compared to the traditional auger plate method. For 50 samples, that could take anywhere between 22 and 81 hours. Flow cytometry is also pretty accurate based on what we've looked at so far. The standard deviation for repeated measures is very small as compared to again, doing the agar plate method, which requires a human to sit and count individual colonies on a plate, that standard deviation is going to be higher and is going to vary from person to person. This is much more efficient because one of the highest costs of doing science is human labor. And you can start the flow cytometer and walk away from it and do something else at the same time. And again, removing human error is very important, and flow cytometry is almost completely automated. So it makes it a good candidate for something to look at in terms of trying to find another way to quantify antibiotic-resistant bacteria. So flow cytometry has been used to look at bacterial viability and vitality. And when I say vitality, I mean looking at cell metabolysis. And so for viability, the way that we're currently using it is using a cyto dye, which is a membrane permeable dye that binds to DNA. So it's going to bind to all cells or anything that has DNA in the sample. And then we also add propydium iodide, or PI, which is a membrane impermeable dye. So if it manages to get through the membrane, that means the cell is dead, because if the cell's membrane is permeable, then the cell is no longer viable. So what we end up looking at is the ratio between all the cells that we see and the cells that are stained with the PI, meaning that they're dead. So if we're looking at metabolic activity, we have a couple of different things that we're going to try. We're going to look at this calcium violet dye, which binds to esterases and lets us know about how quickly cells are reproducing. And bacterial membrane potential, or the potential on a bacterial membrane, is also a way of measuring metabolic activity. So thus far, kind of in the literature, there's been limited use of flow cytometry for looking at antibiotic susceptibility specifically, and there's also been really limited use of using flow cytometry for looking at complex environmental cultures. Most of the bacterial work that's been done thus far with flow cytometry has been looking at one individual species. So this is going to be a little bit different of a ball game, because the samples that we're looking at are highly complex and contain lots of different types of bacteria. So my hypothesis with this aim is that we will be able to put together a method to assess antibiotic susceptibility for both bactericidal and bacteriostatic antibiotics that we isolate, for bacteria that we isolate from soil and water. The first aim that we're going to look at is to demonstrate that flow cytometry is at least comparable in sensitivity and efficacy and reliability to the traditional agar plate method. And secondarily, we wanted to determine that bacterial viability and metabolic activity under antibiotic selection with fluorescent labeling is something that we can do reliably using flow cytometry. So I have a little bit of preliminary data. The figure on the left shows on the x-axis, you've got agar plates, which is the traditional method, and the bacterial concentration in colony forming units per mil. And on the y-axis, you've got the flow cytometry bacterial concentration in cells per mil, and how for the same sample, how they measured against each other. And we had an R-squared of above 0.98, which is fairly good. It means they're tracking pretty closely to each other. So that's a good sign. And then on the right, it shows that the two methods are also tracking, like they're reading about the same concentration, which means not only are they tracking well together, but the lines should be sitting right about on top of each other. So that's important. We've started a little bit playing with the bactericidal method with ampicillin. And so what we do is we set up these kill curves with bacteria that are resistant to and susceptible to ampicillin. And ampicillin is a bactericidal agent. And what we do is we want to see it change. We want to see changes in the ratio with propydium iodide in relation to the ratio that we set up. And so what we set up was 100% susceptible, 75% susceptible, 50, 25, 0. But what you'll see on the line here is where it crosses our y-axis, we would want it to cross at 0, but that's not where it crosses. And what we kind of figured out kind of along the way is that ampicillin lyses cells. So we're probably losing a lot of information along the way. And this was done before we started incorporating the cyto dye. So we're hoping that when we incorporate the cyto dye, this method kind of cleans itself up a little bit and we might get some better results. So the experimental design that we're going to do for this aim, we're going to repeat the bactericidal stuff, continuing to use the PI because that seems to work well. However, we're just going to redo the kill curve experiment, except incorporating the cyto dye so that hopefully we can catch only the bacteria that have not been lysed. And then for the bacteriostatic antibiotics, we're basically going to set up a similar experiment, so a kill curve type of thing, and we're going to test out both of those different dyes, the calcium violet and the membrane potential, and just see which one appears to be more reliable in comparison to agar plates. And then finally, we're going to use whichever method turns out best, and we're going to try it on some environmental samples that we take from the Jefferson Parish wastewater treatment plant. And we're going to compare both methods, the agar plate method and then our methods that we've determined for the flow cytometer, and just compare cost and time investments, compare results, and just see how things shake out compared to each other. So hopefully, I expect to create a reliable and reproducible method for being able to measure antibiotic-resistant bacteria from environmental samples against both bactericidal and bacteriostatic antibiotics. If I just have copious amounts of free time, there's a possibility that we might be able to look at assessing multi-drug resistance within a single sample, just because of where these dyes sit in comparison to each other, where their wavelengths are. That might be a possibility. But one of the limitations is that method development is slow and it takes a lot of optimization, and it's taken a lot of optimization for us to just get to this point. And then my subsequent aims rely on this working properly. However, it does appear that this works at least as well as the traditional plate method, so we're hopeful that we're going to be able to continue using this in the subsequent aims. So the second aim is kind of the largest of the three, and it's to sample and analyze soil and water samples across the human gradient for antibiotic-resistant bacteria and their genes in 2 national parks. So a little bit of background again. Resistance genes have been found in the environment. They've been studied much more in places where we expect to find them, which is the figure on the right. So that is from wastewater treatment, a municipal wastewater treatment plant. And in general, they find them more in the influent than they do in the effluent. And then kind of the further away you get from the source, the less often you find them. But the figure on the left is interesting because these are antibiotic resistance genes that were studied in Antarctica, where they were actually found. And what they found, what that figure shows, is they found the resistance genes more frequently the closer they were to where there was human activity. So the further away you move from the scientific stations in Antarctica, the less often you would find these antibiotic resistance genes. Neither of these studies incorporated the use of antibiotic resistant bacteria, which again tells us nothing about viability. And other than kind of a few studies being done in Antarctica, there have been very few studies looking in pristine environments, and there have been almost no studies other than the one that I've just shown you here that are actually measuring these things along a human gradient. So if we really want to say with some certainty that humans are causing this, then we're going to need to actually look at this human gradient. So my hypothesis for this aim is that antibiotic-resistant bacteria and their genes are going to be present, and they're going to be present in greater quantity at sites with more human activity. My first kind of sub-aim is to go out and to measure these resistant bacteria and their genes in soil and water along multiple transects in Rocky Mountain National Park and Jean Lafitte National Park, which is here outside of New Orleans. The second aim would be to determine the taxa of the resistant bacteria and to determine if the quantity of certain ARGs are present, and then kind of to do an untargeted approach to look at which, just kind of a broad spectrum of which resistance genes are present. The third sub-aim is to quantify the presence of heavy metals in the soils from both of these places. And then finally, the fourth aim kind of ties all of those together, and that's to put together a regression model that describes what of these things might be predictors as to whether or not the genes and the bacteria are present in the soil and water samples. So I have a little bit of pilot data from my master's thesis, and I went to national parks, collected soil and water, and basically the too long didn't read version of what you're seeing here is that it does appear like factors associated with human activity and human density, such as which national park, which county the park is in, the visitation level of the park, farm density in the county near the park, those are all significantly associated with whether or not ampicillin-resistant bacteria were found in soil and water samples. However, the graph on the right shows that it's not necessarily this like really nice trend of whether or not the human activity was low, medium, or high. So it shows that more research needs to be done. So to kind of get into the experimental design of how this is going to be done, on the left you can see Rocky Mountain National Park. It's got several types of ecosystems within it. It's a rather large park. It takes up a little over 265,000 acres. It spans from 7,600 feet in elevation all the way up to 14,000 feet up at Long's Peak. In 2018, they had their record visitation at 4.6 million visitors, which just for the record is a vast underestimation of how many people actually visited the park. Within the park, there are over 350 miles of hiking trails, and it sits right along the Continental Divide. In comparison, there's Jean Lafitte National Park, which is just outside of New Orleans. You can see New Orleans there on the map. It's a subtropical bayou. The whole park takes up about 23,000 acres. Its elevation span is somewhere from -4 to about 10 feet. Its 10-year visitation average for the last 10 years is about 415,000 visitors per year. They have exactly 6.85 miles of hiking trails, and it's pretty difficult to access anywhere within the park that's not on one of those hiking trails because they very much discourage you going out into the bayou, even if it's on a kayak or something like that. And that park is fed almost entirely by lakes, which are fed by the Mississippi River, which I'm sure you are aware of, we are at the bottom of. So at any individual park, the first step is to do transect collection. And since this has already been done at Rocky Mountain National Park, what I tried to visually show here is that I took the park and divided it into 4 quadrants. And within each quadrant, 2 transects were selected, trying to pick one that was a little higher use and one that was a little bit lower use. And then for each of those transects, 30 water samples and 30 soil samples were collected, which I'll talk about in just a minute. And then those samples are preserved and then measured. And we also measure human activity and impact for each of those transects. So water sample collection. So along each transect, it involves hiking out to some predetermined endpoint, which is usually kind of what is thought of as the end of the trail. So the picture on the left is an example of one of those at Bluebird Lake. And if you can find the 2 naked people in the picture, I'll give you $1. They're there. So there were 30 water samples collected from each transect, and there were actually 7 transects for water because one of the chosen transects at Mount Ida ended up having no water on it whatsoever. So we ended up collecting 210 total 1-liter water samples. These samples are carried immediately back to the car where there's an ice chest and then kept refrigerated until the culture assays are done within 24 to about 72 hours. Soil collection is done at the same time that water is collected. And again, we did 30 cores per transect, and that resulted in 240 samples collected. Again, they're collected. We take them immediately to an ice chest back at the trailhead as soon as possible. They're refrigerated until the culture analysis is done about 24 to 72 hours later. So how are we measuring human impact? Well, the way that we're doing it right now is by measuring the distance from the trailhead, and that's operating under this assumption that people are less likely to hike very far. That we're assuming that people are lazy. We also have access to some trail count data, which is kind of what the bottom right image shows. It's basically these cameras that count as people walk by, and we should have some of that information for some of the trails that where samples were collected. It's possible to also measure indicators of human waste, both chemically and biologically, by looking at biological measures. And then again, we're also gonna try to look at heavy metal contamination, since especially in places like Rocky Mountain National Park, where there used to be mining operations. We know that some of those heavy metals that are there are a result of contamination and were not there naturally. Something that is important to bring up is that there are some definite confounders that we don't necessarily have a plan for dealing with yet. And one of those is wildlife. So both the top two pictures, you can see there are tons of elk everywhere. They make these kind of annual migrations that go up and down in elevation. So they're constantly moving around. People are constantly interacting with them, even though they're not supposed to. Again, with the smaller wildlife, like this little guy sat there and was begging me for granola. So humans are interacting with the wildlife are interacting with each other. How does that contribute? That's not currently something that we're looking at. Landmarks. Landmarks might be a confounder in terms of using distance from trailhead as a metric for human activity. So the bottom left picture is a picture of Long's Peak, which is one of the most popular places in Rocky Mountain National Park. And if we were going to consider distance from trailhead as our metric for human density, something along the lines of over 200,000 people climbed Long's Peak this last year. And it's a really long hike to get up there, whereas most people wouldn't necessarily do that, wouldn't hike that far to see something that's not a landmark. So we need to take into consideration on some of our transects what might be landmarks and how that might be confounding it. We also have no measure of what just kind of natural background resistance might be. Physical and chemical parameters, particularly in the water, so how fast the water is moving, DO, other chemical parameters, pH. We have no measure for those in our samples, and then cannot be sure how that might be affecting resistance. We also have no measure for time, or we have measures for time, but we don't know at this moment how time might contribute to how resistance is changing from month to month or from year to year. So the way this analysis is done, we have our antibiotics of interest. Two of them are bactericidal, that's levofloxacin and vancomycin. Those were chosen because levofloxacin is of interest to pathogens that are prevalent in Rocky Mountain National Park, and vancomycin is chosen because it's considered to be a drug of last resort. Our bacteriostatic antibiotics are doxycycline and sulfonamides. Again, doxycycline was chosen because it's a drug that would commonly be used against pathogens that are of interest in Rocky Mountain National Park. And then we chose to use sulfonamides, and then it ended up that we weren't able to actually measure that in the samples that were taken this past summer. So using the auger plate method from the previously taken water samples, we were able to use 114 of them, and we were able to use all 240 of the soil samples. And kind of the way this process works is the water is collected, the water is then filtered, and then depending on which method we're going to use, the bacteria are either put on a plate that's got the antibiotics on it, or in cell media that's got the antibiotic in it and measured with flow cytometry. The colonies are counted, or the flow cytometer does the counting for us. We're going to normalize against total bacteria, so meaning we count how many bacteria are there regardless of antibiotic, so with not looking at whether or not they're susceptible to antibiotics. And then we create this ratio, and we're going to use that ratio to kind of do our data analysis and create little heat maps, kind of like what I've got shown in this picture here, and kind of see how this changes spatially. So there's also some genomic analysis that's being done. I'm first going to talk about the targeted genomic analysis. So we have some resistance genes that are of interest. And again, these genes, the genes we chose go against the same antibiotics that we chose for the culture-based analysis, with the exception of INT1, which codes for a class 1 integron. Integrons are these mobile genetic elements that tend to carry antibiotic resistance genes, and the literature suggests that they're really important for how resistance genes move around the environment. So we're interested in looking at those as well. We're going to do our targeted based analysis for these genes using quantitative qPCR. And again, the way this kind of the cycle of this goes is you go out, you collect the samples, you filter the water, we do a DNA extraction, we run qPCR. Again, we're going to create this ratio to kind of normalize against how many bacteria are there. And then we go and we do the data analysis to see how this changes spatially or compared to any of our other predictors. We're also going to use an untargeted approach, so using metagenomics. So we're going to use DNA from the samples that had resistance, so we can look and see what might be going on in the samples that had interesting resistance profiles. And so that's where we've used that extracted DNA. And then we're going to do shotgun sequencing using an Illumina platform. And then what we end up doing is we create figures that look somewhat like these. So down on the right kind of tells us what different types of antibiotic resistance genes are in our samples. And then on the left, we're able to make these taxonomic trees. What different bacteria are there in our samples and what of them are resistant? And then to kind of look at that from a statistical standpoint, we're able to use principal component analysis and hopefully some network analysis. The third sub-aim of this study is looking at the heavy metals. So we take the soil samples that we've already got, we digest them with strong acids, we put them in the inductively coupled plasma mass spectrometer, or the ICP-MS, which gives us these readouts. And then again, we can do this correlation analysis to where they are spatially to see if there's some kind of correlation between where we're finding these heavy metals and where we find the resistant bacteria and things of that nature. So finally, kind of again, putting it all together is trying to create this predictive model to see if we can figure out what different factors make it more or less likely to find resistant bacteria or resistant genes in any particular sample. So the way I would go about doing this is first we're going to exclude predictors for which we have no data, even though we think they might be important. If we don't have data on them, we can't include them. Then we're going to use methods for predictor selection, which may just be whether or not they're significantly correlated, using like either forward or stepwise selection. And then ultimately we can develop a model and look and see if it has any kind of goodness of fit and decide whether or not it's a good predictor of whether or not we find resistant bacteria or genes in any particular sample. So I have a little bit of preliminary data to show you from the Rocky Mountain study. On the left, what you can see is this is for all trails together combined. for levofloxacin resistance with that resistance ratio that I was telling you about. That's on the y-axis. And distance from the trailhead in miles is what's on the x-axis. And there is a statistical correlation that's negative that says the closer you are to the trailhead, the higher that resistance ratio is, which means you're more likely to find the resistant bacteria the closer to the trailhead, which is where we expect more people to be. And then when you look at doxycycline, we're seeing, and you put all the trails together, again, we're kind of seeing this same trend and to qualify, these are in water samples. However, then when you look at soil samples, this doesn't quite hold up as nicely as it does in the water samples. For levofloxacin, it is still, it's never statistically significant. The correlation when you include all the trails for either levofloxacin or doxycycline. So what I've tried doing is breaking the trails down into halves. So maybe it makes a difference by trail half, which it appears like it might make a difference. But what ultimately we need to know is there may be something different that's happening in the soil. There may be some other important predictor. There just is more work that needs to be done. So there's been a little bit of work done on the qPCR, mainly trying to get all the assays set up and ready to go so that we can run the actual samples. And most of that work has been done working to get the standard curve set up. And so we're using double-stranded DNA that comes from a company called IDT, using these gene blocks as our templates. And so we've been trying to make these templates work in reproducible standard curves. And that's the kind of what I have to show for that so far. Ultimately, with this particular aim, I expect to come up with about a database worth of information because we end up knowing a lot about all these individual sites, these different trails, these different parks. And so it's going to end up giving us a lot of information that we can look at and analyze in a lot of different ways. There are definitely some limitations to this aim. First of all, it's cross-sectional, which means that we're just looking at one moment in time. And as I've said before, I feel very strongly about the fact that time is probably a very important factor in how these things are changing. Also, the comparison of Jean Lafitte to Rocky Mountain might be spurious because the parks are very different. The way people use them is very different. Where they are located, the ecosystems that they're in, just almost everything about them is different. And so trying to compare them would just be a very difficult thing to try to do. So we'll have to be very careful about that. And so finally, to get to my third aim, to remind you of what it was, it's to look at the genotypic and phenotypic effects of 5 different heavy metals on the antibiotic susceptibility of both gram-positive and gram-negative bacteria. And just to give you a little bit of background, heavy metals have been found in soils. That should be no surprise. In general, it's found in places where there's contamination, so near mining sites or agricultural use because they're used in like fertilizers and things like that. There have been some biochemical studies done that have correlated bacterial genes to antibiotic resistance, or that have correlated heavy metal resistance genes to antibiotic resistance genes, and that happens through 2 different mechanisms that this figure shows you. There's co-resistance and cross-resistance. So cross-resistance is when a bacteria evolves something like an efflux pump that is non-specific. That means it can just pump out a toxic substance and it doesn't matter what that substance is. Like it might be arsenic, but it might also be vancomycin. Like the cell does not care. Co-resistance is when you have genes that show up on this same plasmid, and so it gets passed along together. And rather than just one gene getting passed along, both of these genes get passed along together. Again, there have been some correlation studies done that have looked at soils from agricultural areas or areas near mining sites and they look at the concentration of heavy metals, and then they look at the concentration of resistance genes, and they find that there are correlations between them. However, no studies have been done to actually demonstrate that heavy metals are causing bacteria to become resistant. And so that is what I am aiming to do. So my general hypothesis for my first sub-aim is that heavy metals will induce a phenotypic and or a genotypic change in antibiotic susceptibility when the bacteria are in growth media. And so to kind of just put that into a figure form for you, we're going to use E. coli as our gram-negative bacteria and Enterococcus faecalis as our gram-positive. We're going to subject it to a sublethal dose of one of 5 antibiotics, heavy metals. These metals were chosen based on the correlation studies that had been done in the literature. And then we're going to expose them to one of 5 antibiotics. And again, keeping the same antibiotics from the other studies just to kind of for consistency's sake. And then to just demonstrate, we're going to measure the resistance genes of interest, and then we're also going to look at the minimum inhibitory concentration, which is the MIC, which just means the smallest concentration at which the bacteria start to be inhibited by the antibiotic. And we're going to see if that changes after they've been exposed to a sublethal dose of heavy metals. So for the second sub-aim, we're going to look at essentially the same thing, and the hypothesis is that there will be a phenotypic and or genotypic change in antibiotic susceptibility, but in a microcosm, and that's going to happen over time. So to kind of bring it back a little bit, for that first sub-aim, doing it just in cell culture, the first step is going to be to determine the dose response curve for both E. coli and Enterococcus to those 5 selected heavy metals. And we also need to determine what their minimum inhibitory concentration is to those 5 antibiotics. Then we're going to expose them to that highest sublethal dose. We will then expose them to a range of the antibiotics. And then we're going to look and see how that MIC changes from start to after exposure to heavy metals. So again, kind of the way we're going to do that is we've got these bacteria. We're going to expose them to these heavy metals on this huge concentration range, and for figuring out the dose response curve to the heavy metals, or to figure out the minimum inhibitory concentration, we're going to expose them to this concentration range shown in the bottom table for each of those antibiotics. And that concentration range came from the Clinical Laboratory Standards Institute. I didn't just make those up. And then we're going to measure bacterial viability or vitality depending on the antibiotic using flow cytometry, and find the dose response curve. So once we know those things, we can do the actual experiment. So we'll take those bacteria in cell culture media, expose them to that highest sublethal dose of the metal. Then we will expose them to what we know their minimum inhibitory concentration is, and a range of higher ones. We'll then measure using both flow cytometry for the phenotypic change to see whether or not they die or they live. And then we're also going to use qPCR to look to see if there's a difference in change in the number of resistance genes. And we'll look to see if that inhibitory concentration goes up or down, if it changes at all, and if the concentration of these resistance genes goes up or down. And again, these resistance genes that I have listed here, I chose those based on resistance genes in the literature that have been shown to be related to both antibiotic resistance and heavy metal resistance. So that second sub-aim is very similar to the first one, except for what we're going to do is we're going to set up a microcosm, and we're going to watch how this changes over time. So we're going to put E. coli and Enterococcus in something along the lines of like an aquarium with some soil, and measure them first. So take a baseline measurement of what their minimum inhibitory concentration and what their resistance genes are, which theoretically should be 0. And then we will expose them to that sublethal dose of the heavy metals. We will take a measurement at instantaneous and then every 3 days up to 30 days and make those measurements. So using qPCR for the resistance genes and using flow cytometry to see if there's a change in the MIC, to see if there's a change in the actual phenotypic susceptibility. And the reason why we might add this experiment on is because in a microcosm, things behave a little bit differently. It kind of starts to add in the variable of how things might behave in an actual environment as compared to a cell culture environment. So my general expectations for this aim is to figure out whether or not heavy metal exposure alone causes a change in antibiotic susceptibility of bacteria. And we want to determine whether or not that role changes or when you introduce the concept of a microcosm. There are a few limitations here. The outcome is extremely uncertain. There's a total chance this is not going to work at all, that what we're going to find is just nothing. And that's a little scary. But then what we'll know is that there are other things going on out in the environment that contribute to these correlations between heavy metals and resistance genes. We might be excluding, I'm only looking at 5 heavy metals. There are tons of other metals in the environment. There are tons of other metals that are important to antibiotic resistance and particularly in clinical settings. And those are being excluded here. Those might be of importance. Those might be the ones that work. We don't know because I'm only one person and I cannot look at all of them. And also this study is not highly generalizable. Again, if what we want to know is how this is working out in the environment, a lab-based study doesn't necessarily tell us very much about how things are happening out in the environment. So to kind of wrap up, we have discussed that antibiotic-resistant bacteria pose a serious threat to public health, and that both antibiotic-resistant bacteria and their genes are present in the environment. Studying antibiotic-resistant bacteria with the current agar plate method is very costly. Flow cytometry might be able to provide a high-throughput way of measuring them in environmental samples. Characterizing both resistant bacteria and their genes along human gradients could fill multiple knowledge gaps, including how humans might contribute to this and how these things are distributed within localized environments. We also don't know very much about the factors that are associated with the dissemination, and the development of antibiotic-resistant bacteria out in the environment. And so what we're going to look at is, do heavy metals have an effect on the phenotypic and genotypic antibiotic susceptibility of bacteria? And I apologize for running over. Thank you for attending, and if you have any questions, I'd be happy to answer them.
So the section is open for comments and questions. But I kindly ask the committee member to hold your talk and questions later. So question and comments for Laura?
I have a few, Laura. This is quite comprehensive, really, really exciting. You mentioned the things that I was thinking about too. So how you will build multi-drug resistance, because that's really relevant from a public health perspective. Are you any thoughts on that?
Yes. It's difficult to look at the phenotypic level, so looking at the resistant bacteria about whether or not they're multi-drug resistant. With the agar plate method, it's not all that difficult to do. We can take an isolate and expose it to lots of different things. It just kind of exponentially increases the amount of work there is to do. So we're hoping that flow cytometry kind of introduces this ability to do that cheaply and maybe a little more easily. Because if we're finding multi-drug resistant bacteria out in these pristine environments, would be something of concern.
Right. I'm just translating that to humans as well. Now, is it possible, or are you going to look at influences among or between bacteria in terms of resistance?
I'm not sure I understand your question. Can you-
So you're going to look at resistance for specific bacteria, right? But could they, at the genetic level, interact with each other to create more resistance?
Yes, absolutely. And I'm not targeting any specific bacteria in the environmental studies.
Okay.
So we're kind of looking broadly, trying to see who's there and who's resistant.
I see.
But yes, they absolutely can contribute to each other,
That's-
who's resistant. Yeah.
Is that maybe an option for untargeted analysis? Not for your PhD, but I'm just asking you as an area of science.
Part of what we're trying to look at is which of those genes are present, and by which of those genes show up in that untargeted approach, that might tell us, that might inform a little bit on how much of that horizontal gene transfer is going on. Like if we're seeing a lot of those integrons, then that might suggest that there's more horizontal gene transfer going on than some other mechanism of action.
So that I would be excited to know how that's going. And then my last question was on what do you think are the greatest, or which do you think are the greatest differences? Which factors are the greatest differences between the two parts? Is it temperature? Is it, what is it? You think?
I mean, they're just so different. The way people use them, the way people interact with the park is a huge difference. And then just everything ecological, like I'll just put it under the ecological umbrella, would be my second choice. Okay, thank you. Did you have a question, Dr. Miller?
Yeah, about the metals. So why would you not use the same counter ion when you're using lead acetate, mercury chloride, things like that? Is there effect of the counter ion of the metal?
Right, that's a really good question. And so that's something that I recognize as a limitation, that I don't necessarily know if that's the same ion it might encounter out in the environment. And so I can't necessarily account for that at the bench level. Does that answer your question? Did I answer the right
You
question?
could all use chloride salts or
Oh, I see.
sodium salt, or whatever. Different salts use the same counter ion. For all. You use arsenic chloride, lead chloride instead of lead acetate.
You gotta watch the solubility of the chloride salts though. You have to get out of the pH range where the bacteria will actually grow. So,
That's another thing, when you add an acetate, it's going to change pH.
Right, and
So then that's another variable.
correct. And we don't even know that we're going to be in the right valence state for it to be for toxicity of what we might be looking at. So that's part of why there are a lot of unknowns kind of associated with that particular project.
Use arsenic iodide, triiodide.
Yes.
Is there a reason? It's ASI 3. Is that right?
Yes.
Is there a reason why you use that? Because I think iodine has some microbial effects too, right?
I believe so, yes.
You use iodine to disinfect cuts.
I just use them at suggestion. So since I'm not a chemist.
Okay. One more thing about the metals is a good one to try would be silver, because silver sort of double dips. It's in the environment, but it's also becoming much more prevalent use in disinfecting in hospitals and other situations to avoid chemicals, disinfectants in hospitals.
Sure, that's a great point. Yes.
So that would be one I would try. And then the mechanisms are also known, so you kind of know what to look for.
Sure.
Silver and mercury, those are pretty well characterized, the genes that confer resistance to those, so you kind of know where to fish for those.
Sure. Thank you. Yeah.
Laura, I think you mentioned you're aiming to using some temperature data, I believe. And it might have been for your master's preliminary data that you showed. I don't know if it's the land surface temperature data that you were using, or it seemed like there was like an average. Was it a monthly average?
Oh, right, yes,
daily
it
average?
was monthly ambient temperature average for the month in which sampling was done.
So was that documented there at the national park?
Yes. Yeah. For as close to the area of site of sampling as possible.
Hey, Laura.
Hey.
This is Scott from Rocky.
There's no questions. No more questions and comments?
Hey, Laura, can you hear me to ask
Thank you
a
so much for attending. Thanks.
Hey, I think I'm hearing you in the meeting, maybe?
Yes,
Okay, and you're hearing me.
yes.
Great.
I have a question for you.
Okay, give me just 1 second. We're gonna do committee questions in just a minute.
Yeah.
Oh, yes, yeah, of course. Yeah, I'm gonna send, the deadline is on Tuesday, so I'll be making a decision by then. And I'll definitely let you guys know.
I'm gonna move
Good
you.
job, Laura.
Thank you, Sarah.
So Scott, can Scott hear us?
Yeah.
So
Can you
Scott,
yeah.
Scott, if you don't know Scott, he is an ecologist and he's the director of the Research Learning Center at Rocky Mountain National Park. So and he helped a lot with the sampling and the location. So.
Where do you want me to be?
As long as you feel comfortable.
Scott, did you have a question?
I do have a question. Sorry, I wasn't sure if I heard him introduce me, but I wasn't sure if that meant to go ahead and ask away. So the one thing that I really cued in on during your presentation is when you talked about under AIM 2, kind of the dissimilarity between what you found using distance to trailhead as an indicator between soil and water. And I'm wondering if the reason for, and this could apply to water, and so I need you to help me understand this a little bit, but as you move away from each trailhead, both in water and in the soil, you're going to be going up in elevation inside the park, and thus a lot of things are going to change. And one thing in particular would be temperature, and that's much different for soil than it is for water, right? And my question kind of is do you think some of those things like soil temperature, and then additionally things like topographic variability, such as like you're walking along the trail, you're collecting at your kind of intervals, and all of a sudden you find yourself in a cold air sink or a north-facing aspect versus a south-facing aspect, would have impacted why distance to trailhead didn't show to be a good indicator for soil over water?
Yes. The short answer is yes. So I did look and check to see if elevation was significantly correlated with either soil or water. And what's interesting is it's counter to what we just said. It was significantly correlated with finding it in water, meaning I was less likely to find resistant bacteria at higher elevations in water than at lower elevations, but in soil, the elevation was not significant. But
and that leads me to believe that maybe something like, well, there's always soil properties versus water, but that could be a temperature indicator because, surface waters that you're measuring are going to be warming under environmental change at a totally different rate than what we're seeing in soils. So you got this low elevation soil that's warming maybe at a different rate than high elevation soils and vice versa, for water. Just food for thought. It was just one thing that popped into my head of like, oh man, how is temperature messing with this and the rates of change? And using elevation as a proxy for that might be something to just consider.
Yeah.
The other thing that came to my mind is like, I don't know how these ARGs and ARBs really interact with soil anions and cations and some of the other soil properties that are going to be changing with elevation, let alone you probably went through 3 or 4 or even more different soil types as you walked along some of these trails. I mean, many of the trails and sites you selected were, from start to finish 6-7 miles one way. So a lot of variability in there and wondering if that could help understand why you're seeing such a messy signal between the kind of two variables or the two sampling types.
Yeah. One of my plans is to use the USDA web soil survey for soil texture and see if perhaps soil texture might have something to do with it.
Yeah, that's definitely going to be the best data set that we have available without just taking your samples and analyzing them for that themselves. Right?
Which
so.
would take forever.
And thank you.
Yeah.
And great job on the presentation, by the way.
Thanks. No, because I didn't have a probe.
Yeah, so did I hear somebody ask about soil moisture?
No, you asked me about water temperature, if I measured water temperature, and I said no. I did start measuring soil pH at the beginning of the season, and then my pH meter broke in half, so I stopped.
Classic. The other thing, though, depending on, we can look through, we might have a ton of temperature loggers out there in our streams.
Okay.
And I can look at the partnership that we have with Fish and Wildlife Service to see which streams you sampled and the location for the loggers and get those raw data potentially to address that water temperature question.
Great. Yeah, that would be fantastic.
So, any comments, Dr. Lee, Dr. Wickley, Dr. Wilson? Questions?
So this is really ambitious. It's a lot of work. How much of this is not even started yet? Just give me a rough percentage.
We have all
The third aim is essentially unstarted.
That's the one I'm probably most concerned about.
Yeah. All the other, aim 1 was probably 60% done. Ish, yeah. And aim 2, depending on as is 35% done, just because the in-house work hasn't been done.
Well, my feeling with the third aim is it is almost its own dissertation. When you have all the microcosm work, all the non-microcosm work with all the metals, with all of the antibiotics, I'm seeing 5 by 5, and then times 3 for replicates minimally, and you might have to repeat some things.
Yeah.
Chuck actually brought up a lot of things I was thinking of when it came to the metals and just their chemical forms.
Right.
Which at some point you're limited because that's what's synthesized by a chemical manufacturer that you can get access to. And as Mark mentioned, some of these things are soluble unless you drop the pH down to 5, and then your bacteria die anyway. So my recommendation, honestly, is I would scrap the microcosm work. I mean, that's going to be a lot of work. I'm not sure. I mean, it's something you could pursue in the future, certainly. I just think for you, it would probably be best if that were left to another time or another person.
Sure.
And I see some kind of inherent issues with that. You sort of said that you expect your background resistance, or you're setting this up in a way that they're going to be, there won't be any resistance. Was that the idea? What it sounded
like from starting in the
so
beginning.
you're trying to evolve this resistance across all these different things. The way natural selection typically works is the resistance is already there. It's a part of the genetic variability that's in the fabric of the population. And so then you add this selective factor, and then that group is the one that tends to go on.
Right.
Theoretically, if you had a population, a completely isogenic population of bacteria that you put under this pressure, you're going to kill them all.
Yeah.
Right?
Yep.
Because the ultimate source of variability is mutation. And I don't think that's your experimental design does not really allow for that.
Okay.
So if it were me, I would consider not including that, because I think your dissertation is plenty rich without it. Including that third aim where you're actually doing the heavy metals and looking at the resistance and that kind of stuff, but in the absence of doing that microcosm work.
Yeah.
So that's really other than that,
Yeah, we kind
it's
of
a really nice.
realize that, especially the work involved very intensive field work, and it's going to generate a lot of data.
It will.
So
Be a mountain of data.
unless you want to spend 3 more years here.
And like I said, it's still a project we're doing that you
yeah.
can pursue, but I just think for your dissertation, I'd take it off. That would be my suggestion.
I would second that.
Okay.
And a lot of that's like you did a little bit of the preliminary stuff with the metals already. And you saw
found
exactly
that.
what you're talking about. When you use a ATCC strain, that's everybody just dies.
Yeah. And that's
Right.
It should.
work,
Which
right?
makes sense. Right.
Yeah.
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 That was my only thing. I think it's solid. I know you guys have been working on this for a while, so I'm really not surprised. It was well put together. I did read over your entire prospectus, and I think with all the comments I saw in there, I was like, all right, pretty well covers it. I just had the issue with microcosm.
Okay.
You get so many people to show up. Like, that's incredible.
The best attended prospectus
I've ever
I have
seen.
seen by far here.
What'd you do? Just hanging things up, or is it word of mouth, or what? Geraldine does that every time, and it's never when we needed more seats.
She did advertise it in the elevator.
She did that. Though.
Yeah, which is not something I'm sure Scott's going, what the hell are they talking about?
There were a bunch of people here.
Yeah,
Well attended.
Yeah, it sounded like it from what I could hear clapping wise.
I was clapping for myself.
So I got a question about the metagenomic stuff.
Okay.
Is that only going to show you bacteria, or is there a way to structure it to where it would also show you what fungi are present that are capable of producing specific classes of antibiotics?
Yeah, it was sure.
So my understanding is that we can, like, when we get the reads back and we run it through BLAST and do the comparisons, if there's a known fungi that we specifically want to see if it's there, then we can look for that. That would be my answer. So yes, the short answer is yes.
So like you'll get 16S from penicillin species, and it'll be able to say that this is a penicillin mold. And then because that mold is there, there's a possibility that it's producing an antibiotic and providing a selection pressure in that environment. So if you can do that spatially too,
then that might tell us something about the soil.
Yeah,
Because
right.
we're also more
Yeah.
likely to
Goes
find
back
those
to
things
some of
in the
those-
soil
Yeah.
than in the water.
Yeah, and
Yeah.
you know, you've always got fungus in the soil. So if they're naturally occurring selections, it would be nice if you could kind of identify.
So actually for that one, I'm not even sure. I think it depends on what type of thing they start with, and kind of like
That's
because
true.
they are measuring the genomic information when they do start with the PCR. I think the template is based on the bacteria. That's my guess.
But it's total DNA.
Right, I know
it's not
total
just
DNA,
bacterial,
but,
it would be fungal too.
I think they are looking into a number of specific gene or protein type of things.
For the qPCR, but for the metagenomics, if we're sending them in just extracted DNA, then that would still be total DNA.
Okay.
Yeah.
Okay.
Yeah, they're just sequencing that.
Right.
Right.
Putting it up into a library,
Yeah.
sequencing all of it.
You are not doing apricot sequencing, you just basically shotgun sequencing.
Right.
That's gonna be a massive thing.
Yeah, that's gonna be an informatic nightmare.
You're gonna have stuff in mind for years to come. Oh, that'd be good.
So how are you gonna regress one national park that's in the Rocky Mountains versus one that is south of New Orleans in any kind of, like what's the point?
Scott, can you hear that question?
I kind of heard most of it, but could you repeat it?
So if one of the sub-aims had a aim to develop a predictive model based on some kind of regression comparing Jean Lafitte Park to Rocky Mountain Park. But given the just frank environmental differences in temperature and elevation, and one being under the influence of the Mississippi and the other not, like how, I just don't see what kind of information would be garnered from that comparison. So
Yeah, I agree completely, and that was one of the comments I provided on the prospectus back was, with all those variabilities between each park, I don't think you can make a fruitful comparison. I think within a park, probably worthwhile, but among the parks, definitely not. And I think that's kind of for, Laura, you to either say I agree with that, and you kind of did during your presentation, that you said it was, spurious, because the second we try, we've left ourselves open for lots of questions and probing because of the vast difference. I mean, behavior of the people inside the parks, let alone all the environmental factors,
Yeah, like you said, you didn't
I think
water
make that impossible.
profile.
Right.
And the water chemistry
Yeah.
really, and it depends on the time of year you go to John Lafitte.
Yeah.
Yeah, I mean, the only other thing I was thinking about that I wrote in my comments as well was like you could do like headwaters versus sink waters, because we are at the headwaters and the east side of the park where half of our sampling is, does drain into the Mississippi eventually. But even that's like, I don't know if I would feel comfortable making that kind of comparison even.
It just goes through to me, if I were able to kind of start fresh, I would want to go if we're going to do more data collection, which is what would be required as is, like with Jean Lafitte, rather than taking data from Jean Lafitte, I think I'd rather just go back and take a subset of the samples that appear from the transects or wherever that appear to be interesting at Rocky, and then have a year-to-year study and see how it changes, or see how it changes from month to month, like choose fewer sites and see how it changes from month to month within the summer or from year to year, and have be able to incorporate that time aspect.
Yeah, I think that would be very cool in the sense of, the temporal change aspect. But when you think about, the two different sampling, like between water and soil's got a big turnover rate, but not nearly as impressive as the turnover rate of a lot of the parameters in water, right? So like you said, pH, you wanted to measure that. I mean, that's going to change a lot throughout the season, throughout the day, throughout the month. And it would be really cool to have that less of a cross-sectional look and more of that longitudinal or, temporal scale look, in my opinion.
I didn't put down my comments for these questions, but I'm not sure I fully understand the whole structure. One is about general comments. One is general comments. I have other comments here. I'll send to you next week. But anyways, one is I think the specific part description is kind of like, to me, is very unclear.
Okay. Yeah.
I'm not sure how clear you want to be, but basically there's no. For each of them, I think it should be better to have pretty clear description what type of method you are using and how you are doing that. And that's just general things. Another one is basically all related to the metagenomics. One is particularly related to the topic, like the dissemination. So throughout this writing, I do not see much about how you handle this, how you infer specifically what do you mean dissemination. To me, it's more like how these kind of spreads, or it's just you give a description about different locations.
So it all ends up being this inference based on people, I think. And so just the more often we associate it with people, then I'm associating people with the dissemination.
Okay.
And then if we were able to incorporate kind of that like fungal metagenomic data, we might be able to rule that out, or either way, or figure out quantitatively how much it's contributing, which again would be another kind of reason to work in Rocky again, because they have some elk fecal data that we might be able to incorporate and then have this measure for wildlife. So that's more of where I'm trying to go with dissemination. So figure like who is contributing and how much are they contributing.
So that
Okay.
makes sense. But another thing I was thinking that like in viral studies, HIV, whatever, those infectious disease, they usually have kind of like they may have isolated origins, multiple origins, but they may also have like just starting from here and then spread out. So there are papers out there how HIV spread over the world. So based on the sequence data. So I was thinking, since here, I'm not sure about how many origins you may have for those kind of resistant bacteria, whether it's isolated or just bring into a park by different people, or because you have streams, you have winds, you have animals inside, so they brought into the park here, but they spread out by the stream, by the animal. So I think by looking into the genetic data, you may have the possibility just to see HIV. They look into the sequence.
I think that's a great idea.
Based on that, then they see the kind of relationship, whether it's close related, and there is further far, so you can get a kind of like a map type of thing to see whether you have spread, not because of people bringing in, but because once they are there, they spread over the park. I'm not sure whether it can be done, but I think that's one thing to think about.
Yeah.
And also, I think I put comments. I'm not sure whether you want to have some kind of GIS mapping just for presentation. You can put it there and give people some intuitive idea.
Yes, definitely. Yeah.
And also, like for the metagenomic data, because you have mixture of different streams within one location. How many data points you have for this type of thing?
So 240 plus 210, so 550.
So for that, I think what you can do is you may look into I think it's, mention about the interspecies kind of like what's called interspecies interaction in terms of the resistance. So because what I'm trying to say is, in this location, you may have more of this string. So string A, you have 80%, 70%, 20% is string B, and the other place you may have the other way around, 20% string A, 20% string B. If you have a lot of these kind of things, just you have same type of strings, but all have different kind of like proportion at each location. I think statistically you can look into this kind of interaction stuff, because by adjusting the amount, if there's no interaction, then each of them will have just their proportions. You can see that. But if there's interaction, then if you solve the equation, it should not be like a direct solution. You should see the difference. That's, well, at least in statistical sense, that could be the interaction, but it all depends on what type of data you may see there.
Sure. No, that would be really interesting.
And also,
Yeah.
I think that they are asking about different factors.
Right.
That's also why I saying, I said that statistically, if you write it more clearly, then we can see how you analyze them. Because if you have a lot of data points, you can try to put these things together and trying to, because they can be confounding. Like you have elevation, you have soil type, you have temperature. If you can put them into the model together, then sometimes you do have a way to decompose their contributions. It's all, I guess, it again depends on
Right.
what type of data points you have.
Yeah. That sounds great. Yep. That makes sense to me.
So Laura will address all the comments and revise the report and send it back to everyone.
Do I have to leave while you all talk? Is that the thing? No.
We do that for the perspective.
Usually,
yeah.
Sure. Anything anybody wants to say without her here. If there's nothing anybody wants to say or discuss, or do we need to discuss more? Scott, anything else?
I feel comfortable.
I'm fine. Okay, fine. Usually there's a
Piece of paper.
does she need to make all these changes and then we sign her form, or are we comfortable signing her form now? And then she can make the changes and revise. I can sign the form now.
Well, I don't have the form,
Should
so
always bring the form. Hurting these cats. All right, so
Why don't Laura make the changes and revise it and resign?
so I want to make sure to be clear before I make the changes and then make changes again. So we're going to scrap aim 3 completely, and bring it down to two aims.
I mean, you have
That-
so many sub-aims in
It-
aim 2,
yeah,
and
That-
now
that's
you're
not
adding more
Yeah.
with the that are probably going to be a lot more informative
Okay.
than the meta
Yep.
stuff. So, even if it's 2, since 2 is so big, I don't have a problem.
Okay. So scrap aim 3. Then and
or
then
just put it off for another
sure.
day.
Sure.
Another student.
Yeah. Are we in agreement about eliminating Jean Lafitte and going back to Rocky?
That makes sense to me.
Fine with that.
I agree with that.
Okay. And then better description of the current.
So I think that's
Yes.
one should not be like a contingency for signing that, my personal opinion, because those things can take quite some time. and also it's evolving. Like
Yeah.
the statistical analysis part is depending on what you need to do.
Right.
Yeah.
What your data looks like.
Just keep this in mind.
Okay, but a bare minimum, a better description of the statistics I already plan to use, right? Okay, does that cover the changes that need to be made?
And address some of the specific comments.
Right.
Yeah,
Yeah,
that are
yes,
already
correct. That are already there. Yeah.
And is the one on Box the most current version, or is the one that you sent via email the most current version?
The box.
Because you commented to a file on Box.
Yeah, I comment on the file from Box.
And then you
I-
sent one
I think that's a big version.
that's a later version.
I
Okay.
think someone Scott sent
Scott
a
has that.
version through email.
Oh, maybe
with
that's
the-
where that came from.
Yeah,
'Cause
so
he couldn't get it. That's
right, but I haven't made any change. Like since I put it on Box, I have made no changes.
So
Like
that's the
it is
most
the only changes that have happened have just been people's comments.
Yeah, so I just downloaded it from Box, did my changes in comments or made my comments in Word, and then sent that to you directly, Laura.
Yeah, and that was totally fine. I'm just saying I have not made any changes since I sent it to everybody.
Okay.
Actually, I rewrote it to send me to Disney World. Thank you for agreeing. Have a good day.
But I agree with a better kind of layout of the statistics. That was something that was kind of in the back of my mind, but I didn't really want to bring it up to the forefront as I wasn't sure what level of detail we wanted to get into here.
Okay. Is speaking of GIS things, going back to that, is that something you might be able to help me with, Scott?
I think so. So we can kind of talk about that in a little bit more detail, but I think that's something that we can probably figure out. And if not, I can also tap into our GIS program to help sort some of those things out. Use some of the park resources for that.
Sure. Okay. Just checking.
Yep.
Oh, great job.
Thank you. Thanks for coming. Thank you for
Okay.
your time.
Yeah. Cool project.
Thanks.
Are they going to
Oh, you're going this July, right?
Yes.
She's offering.
I'm teaching a class.
Course in Rocky Mountain. Is that mostly college students?
Scott, have you ever been to one of those conservancy classes?
I have.
What's the typical demographic?
That's a good question. It can be highly variable. But if I was going to throw out a demographic, I'm going to say like mid to late age, educated white folks.
Okay. Retired people.
Yep,
Yeah.
for sure. But like I said, it's really hard to know. I mean, with 5 major universities here, and we send those class lists out pretty widely. You never know. You might dredge up some university professors that are like quite interested in this. I know there's like a bunch of people at CSU that are looking not at antibiotic resistance genes, but more of like what are some of those other human impacts in our waterways, and this is going to just add like a little bit of context and value to what they're doing. So you might even pull some of those folks in.
Cool.
Okay. Well,
And
then,
you're doing that as part of your. Go ahead.
I said thank you, Scott. Yeah.
That was it.
Okay, perfect.
Alrighty.
All right. Thanks for everybody.
Thanks.
I'll be in
Yep.
touch with you, Scott, then, because we'll need to make some plans.
Yeah, that sounds good. And then get me whatever forms I need to sign. And if that works for you guys, for me to just print them, sign them, and scan them and send it back, let me know.
So you just don't want me to forge your signature all the time? Is that what you're telling me?
I mean, if you tell me if you want to do that, as long as you let me know, I'm fine with
Okay.
it.
So is that a week of timeline to address the comments sufficient? How do you feel?
I can do that in a week, I think.
We'll aim for a week, and if it goes 2 weeks, all right.
Okay.
Oh,
I like that.
I will be out of country actually on
Oh,
the
okay.
May 7th.
On May
So
7th. So,
we still have some time.
okay.
So if I'm out of country, when you bring up, I think I can just sign it electronically.
Okay.
I have no problem with that. Or I can just sign before I leave.
Okay. That
I
works.
believe you have everything now.
Okay.
If not, you will have them in the near future.
Okay. Great. Thank you.
Yeah, to get the form signed as soon as possible.
Okay.
Get that out of your way.
Okay.
Without that form, you can't.
You don't
Register
you don't
for-
advance to candidacy.
Yeah, right.
I still
So
gotta.
keep you from paying fees,
Right. Yeah. Okay.
That's the real game, which is really important.
Yes, it is. That's correct. Like.
But yeah, I think, I mean, if everyone is okay with signing the form,
Just okay. Thank you, Scott.
Thank you. I appreciate it. Great job. I look forward to seeing how it plays out.
Yeah, me too. I'll email you soon.
Still want to have got so many people in here.
So if you have a question about those kind of related to the metagenomics, I should be in town for
Okay.
2 weeks. Just stop by.
Yeah.
But if I will be out of country for almost 3 weeks, we'll come back May 30th actually.
Okay.
So send an email if you need, and afterwards I should be here for next June, July. So
Sure. I'll try to find a time to come meet with you next week,
Okay.
and we can kind of go through it
okay.
because that would be good for me to kind of just
But
talk
if you
through
want
it.
to, add those things. I'm not sure whether it's doable, but I think it should be
Yeah,
very
yeah, that's
interesting. She added
That sounds really interesting.
because
so.
that's kind of a routine, not necessarily routine, but it's really popular in viral study.
Yeah.
So and also this microorganism study, they're trying to see how where is the origin is and how they are spreading out.
So do you do a lot of microbial metagenomic work, like try to do some network analysis?
So I did some, but not a lot. But if you talk about just data analysis for that,
Yeah.
so let me know what
Yeah,
you can
definitely.
help. And I have I did a lot of pathway analysis actually. But and also I'm doing have a number of collaboration with primary center.
That's great.
All kinds of all new data. And some metagenomics. Okay. A lot of things are
Everything is together, yeah. Especially with your bioinformatics
Right.
skill set, very helpful.
so
Great.
just, any question, let me know.
Yeah, that's great.
And I think it's the topic is very interesting, and a lot of things I don't understand actually. But I think it's very interesting, and especially it's in national park. I like tribal.
Right?
I mean, I've been in Rocky Mountain I think 3 times.
Yeah.
Winter, summer, and long time I think in the fall.
Oh my goodness, it's beautiful. I love it there.
Different
So
seasons have different things.
it's total. Yeah, it
And
changes
just
quickly.
Winter cold, we like climb up to the Bear Lake
Oh yeah,
twice.
yeah.
First day and second day, and chasing. Well, they have frozen lake there, and
Yeah.
although the thing is that I lost my plastic bag, and
Oh,
I
no.
tried to chase it down. Well, it's more like run across the whole lake.
That's really dangerous.
Well, it's frozen, so it's not dangerous for that, but more just like a high elevation. I
Yeah,
cannot breathe.
yeah, that's true. Even at Bear Lake, like
So, but nevertheless, it's just I think it's let me know if I can help.
yeah,
Yeah,
absolutely.
Yeah,
Great.
that's yeah. Of course, we will get you
Sure.
support because otherwise, because you are part of the community. So,
Okay.
Yeah.
So if you have any
oh yeah,
problematic thing I can help, just let me know.
yeah, sure.
Should be here around most time.
Well, thank you so much.
Okay, sure.
Yeah.
See you around. Yeah.
Well, that's